Okay, so let's move on to chapter 4. So the first thing we discussed in chapter 4, okay, enzymes as catalyst. Okay, so a catalyst is something that makes a reaction move faster. Okay, enzymes are considered organic catalysts. Okay, or biological catalysts. Okay. Ribozymes are also considered catalysts as well, and they're sometimes considered enzymes, but they're made out of RNA. Okay. Okay, all catalysts have the same properties. They increase the rate of reaction. They're not changed by the reaction at all, so they can be reused. Okay, they do not change the nature of the reaction. So you start off in the same place, you end up in the same place. You just make it happen faster. Okay, and you lower the activation energy of the reaction. That's what makes it go faster. Okay, so activation energy. Okay. 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 So the activation energy is energy that's required for the reaction to engage in reaction. Okay. So if I have a pile of chemicals, okay, let's make it easy. If I have a pile of sugar on my table here, okay, that pile of sugar will have energy. Each molecule of sugar will have a different amount of energy depending on whether it ran into another sugar molecule or not. Okay, but most of those sugar molecules by far will lack the activation energy needed to break down into CO2. Okay, so if I leave sugar on my table, it will eventually break down into CO2 and water. Okay, it will just literally take a million years. Okay, because very few molecules can get to that level of activation energy. Okay, so okay, adding heat increases the activate. So adding heat increases the energy of the molecules. Therefore, some of the molecules will have the activation necessary. Okay, that's why one of the things to make a chemical reaction go faster is simply to add heat. Okay, but to make chemical reactions happen fast enough, you would have to increase it hundreds of thousands of degrees. So that's not typically a useful tactic when it comes to cells. Okay, heat catalysts can help the reaction occur at lower temperatures. So this is what I mean. Okay, so here, okay, is the average energy of the reactants. Okay, and only this many reactants have enough energy to go through. Okay, and I don't think this really comes close to what the reality of what it is. The reality of what it is maybe that much of the reactants have enough energy to go through. Okay. If you add enzymes, now you have this much of the reactants able to go through. Okay. So you're lowering their activation energy. Another way to think about it, okay, is you need to get over this hill. Okay. And that's the way I usually think about it. So you can be in the first floor of a building and you want to go down to the bottom floor. Okay, and going down to the bottom floor is easy because you can do it, but you need activation energy. So here's the two different activation energy. Without the enzyme, you have to run up 100 stories and then come back down 101 stories. Okay, most of you guys will not have the energy to run up 100 stories. Okay, or you can run up 2 stories and then run down 3 stories, and that most people will have the energy to do. And so that lowers the activation energy. Okay, do you start at the same place? Yes. You end at the same place? Yes. Is it easier? Yes. Is it faster? Yes. Okay. Okay. The function of enzyme is dictated by structure. So each enzyme has a characteristic 3D shape, okay, with pockets that serve as active sites. So really, an enzyme is a big blob that has active sites on it. The active sites are what's important, okay? The reactants are called substrates, and they fit into the active site, okay? The original model was somewhat of a lock and key model. You insert the lock, the key fits the lock, and you insert the key and you turn the lock, okay? The initial fit model doesn't really work. The induced fit model does, because so the induced fit model works like this. You shake hands, okay? Two people shaking hands. I know you guys haven't seen that in like months, but I'm sure you can picture that. Two people shaking hands. As their hands approach each other, their hands have to change conformation to fit into the other person's hand, okay? If you use your wrong hand to shake hands, it doesn't fit, even though your left and right hands are pretty damn similar, okay? Okay, when a substrate binds to the active site of the enzyme, it forms temporary bonds that weaken the original bonds that the enzyme is trying to modify and allows them to break or allows them to form or does something to change the formation, okay? All right, new bonds are formed between substrates that are brought close together by the enzyme, okay? The temporary enzyme substrate complex, okay? Blah, blah. Okay. The amount of enzyme in a sample of fluid can be measured based on the rate of product synthesis. So if you have one enzyme, that one enzyme can make 100 molecules a minute. So if you measure 200 molecules in 1 minute, that means you have 2 enzymes. If you measure 1,000 molecules in 1 minute, it means you have 10 enzymes, okay? All right, the first enzymes discovered were were given arbitrary names like trypsin, chymotrypsin, okay? Later, we decided that was a dumbass idea, and now all enzymes end up with the suffix ase. Okay, we talked about things like helicase, polymerase, reverse transcriptase, all of them end in ase. Lactase. Okay, okay. They also decided to make the first part of the name apply to the function. Phosphatases remove phosphate groups. Okay, kinases add phosphate groups. Synthetases make stuff, okay, make stuff bigger usually. Hydrolases break stuff up, okay. Dehydrogenases remove hydrogen, okay. Isomerases, okay. Isomerase makes different isomers, so you rearrange the atoms. Okay, isoenzymes. Because names are given to enzyme based on function, an enzyme that does the same job in 2 different organs has the same name. Okay, so they're called isoenzymes. Okay, so you have a amylase in your mouth called salivary amylase. You have another amylase that's made by the pancreas called pancreatic amylase. Okay, so those 2 are isoenzymes. Okay. So you can see that here. Okay. enzyme activity, okay, is measured. The activity of an enzyme is measured by the rate at which the substrate is converted to products. It's influenced by temperature. Okay, at first, when you increase the temperature, the enzyme is functioning faster because the molecules are just moving faster, so the substrate runs into the enzyme more quickly, and therefore it increases. But as you increase, eventually what will happen is the protein, the enzyme will denature, and if it denatures, then there's no active site. If there's no active site, then you have no function. Okay, pH. The pH is very, very sensitive for both enzymes, and altering it just by a little bit will stop the enzyme from working. Okay. Okay. The concentration of cofactors and coenzymes. You don't know what those are yet, but I'll get to them in a little bit. Okay. The concentration of enzyme and the concentration of substrate. Okay. So, you know, if you add more enzyme, obviously the enzyme will work faster. If you add more substrate, same thing, because the enzyme will run it. Like if you add in more substrate, the enzyme will run into the substrate more often. There's an exception to this where if you maximize out on the substrate and the enzyme's working as fast as you can, then adding more substrate doesn't do anything. Okay. Possible stimulatory and inhibitory effects based on enzyme function. We'll get to those in a little bit as well. Okay. Okay. So we're studying human physiology. Okay. So human physiology is 37 degrees. Okay. So our enzymes are designed to work at 37 degrees. Our enzymes will work a little bit above 37, so 38, 39. Okay, it hits 40, things start changing. At this point, the enzyme starts to lose its tertiary structure and can become denatured, okay, or basically misformed. You go too high above 40, it doesn't go back. Okay. See, enzymes work ideally at 37 degrees here. That's the top. When you increase a little bit more, they start to work, and then you see a very, very sharp decrease. Okay. Okay. Enzymes exhibit peak activity within a narrow range. That's called the pH optimum. Okay. So, okay. The enzymes in your stomach will function at pH of 2 because that's what the pH of your stomach is. The enzymes in your saliva will function at pH of about 4, 5, 6. Okay. The small intestine will have a pH of about 8, 7 or 8. Okay. Okay. So pepsin 2, salmonella amylase. I was wrong. It works at about pH 7. Trypsin works at even higher than that, pH 9. That will be closer to 8. Okay. All right. Coenzymes and cofactors. Okay. So coenzymes are vitamins, usually. Okay. Cofactors are minerals, usually. Okay. Okay, so enzymes need cofactors and stuff to increase function. Okay. Okay. Okay, enzymes, particularly in the digestive system, are often producing an inactive form called a zymogen that is activated when needed. Okay, so the reason why you want to do this, the enzymes in the digestive system are going to digest things. The last thing you want the enzyme to do is to digest the cell that's making the enzyme. Okay, so what you want to do really is you want to put a pin in the grenade. Okay, so you make this thing called pepsinogen. And pepsinogen literally means pepsin generating. So this molecule is pepsinogen. Pepsinogen is pepsin with a big pin that prevents the pepsin from working. Now you have a separate enzyme that breaks the pin, or use acid to break the pin, and off you go to the races. Okay, so you have one cell in your stomach, okay, called the chief cell that makes pepsinogen. Okay, another cell called the parietal cell that makes HCl. HCl, when you add it to pepsinogen, will make pepsin. Okay, that way you you don't turn it on until it's necessary. Okay. Okay, so as the substrate concentration increases, the rate of reaction will increase until the enzyme becomes saturated. Okay, so the more the more substrate you add, okay, the reaction rate will go faster and faster until it reaches saturation. So by saturation, it literally means every enzyme in the solution is literally working as fast as they possibly can. They're literally like running into substrates as soon as they let go of the previous one. Okay. Okay. Sometimes a single action enzyme can drive a reaction in two directions depending on concentration substrate and product. That's something called the Chatelier principle, actually. Okay. If you get more A, you automatically make B. If you get more B, you automatically make A. It's kind of weird. Okay. Enzymes, okay, usually don't work by themselves. They usually work in long pathways called metabolic pathways. Okay, they begin with an initial substrate. So in this model is the blue thing, and then you have enzyme one, and then you have a whole bunch of intermediates. Okay, so 1 through like 10 or something to get to the final product. Okay, and enzyme works at every step. Okay, sometimes the pathways are branched. Okay. Okay. One of the things we learned was homeostasis and negative feedback. The end product inhibition is the negative feedback mechanism for enzymes. Okay. Okay. So one of the things that you guys probably understand is something called cellular respiration. We burn sugar and we turn it into ATP. Okay. So this pathway is always there, but you don't want to always convert all your sugar to ATP. For one, you need some sugar. For two, you need the sugar to do other things like make fat. And so you don't want to take all your sugar and turn it into ATP as soon as you get sugar. So what you need to do is to stop that pathway. And so how do you know when to stop? Well, when you have a lot of ATP, you don't need to make more. If you don't have enough ATP, you do need to make more. Okay, so the end product is ATP. So ATP will come back and inhibit one of the enzymes. And this process is called allosteric inhibition. Okay? Okay. So here, okay, we have two pathways. If we have too much of F prime will plug into enzyme 3' here and block this pathway, and therefore you'll get this pathway. Okay. Okay. So let's show you a simple pathway first. Okay. Here's my pathway, right? If I'm missing enzyme number 3, for example, okay? If I delete enzyme number 3, one of the things that will happen is, hold on one second. Okay, so if I delete enzyme number 3 that I'm showing here, what do you think I will see? Okay, so I'm guessing you guys figure out that I won't see the final product. I'm also guessing some of you guys will realize I'll see a lot of B, or sorry, I'll see a lot of C. Okay, I won't get to D. I'll have a whole bunch of things that end at C and stay there, and doesn't you know what to do with it because it doesn't go anywhere. Okay, and you're right. And missing the final product F, especially when you need it, is a problem. And this is one of the first things that scientists figured out. Okay, and this is the inborn errors of metabolism. Okay. Okay. So disease occurs due to loss of end product or the accumulation of intermediary products, okay, or alternative products and enrich. Okay. Okay. So here you're missing F. Okay, and then you'll have too much F prime. Okay. Okay. And you can see some of the errors here. Okay. All right, for those of you guys study physics, these are the laws of thermodynamics. Okay. First law of thermodynamics: energy cannot be destroyed or created. Okay. Only transformed. Okay. The second law is that transformation is never 100%, and you end up with heat. Okay. First law of thermodynamics. Okay. Second law: energy is lost with the transformation, and so it increases chaos or entropy. Okay. All right. There are two types of reactions: exergonic reactions that release energy, endergonic reactions that require energy to be inputted. Okay, so chemical reactions, endergonic reactions, are chemical reactions that require input of energy. Okay. Okay. In an endergonic reaction, the products contain more free energy than the reactant. Okay, so if I was making a glucose molecule, okay, from carbon dioxide and water, the glucose molecule has more energy than carbon dioxide and water. So I have to put in the energy from somewhere else. Okay. Okay. Exergonic reactions are chemical reactions that release energy. Okay. So if I was taking glucose and breaking it down to carbon dioxide and water, I produce energy. Okay. We use energy from exergonic reactions to power endergonic reactions inside the body. Okay. Okay. So here, okay, this is glucose breaking down into water and carbon dioxide. We can break this out all in one step, and that would be combustion. That's what happens when you set glucose on fire. That's not particularly effective. Okay, one, that burst will be too much energy. Okay, so what we want to do is extract the energy slowly, one step at a time. Okay. 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 A calorie is the amount of energy a molecule can release. Okay. 1 calorie is the amount of heat required to raise the temperature of 1 millimeter of water to 1 degree Celsius. Okay. 1 cubic centimeter is 1 milliliter. Okay. The calories in food are actually 1,000 calories. Okay. So in other words, 1 calorie of food is the amount of heat required to raise 1 liter of water 1 degree. Okay. Okay. Energy from the environment is broken down in exergonic reactions to drive endergonic reactions in our bodies. Okay. So we break down the glucose, the reactions are exergonic, and we store those reactions, energy from those reactions in ATP. Those ATP will be broken down whenever we need to power an endergonic reaction. Okay. Okay. So how coupled reactions work? Okay. So it isn't like we store a lot of ATP. We don't actually. We usually use them at the same time we make them. So one thing, like these occur at the same time, right? You realize both wheels move at the same time. Okay, they're coupled. Okay. Okay. So this is ATP. Okay. So here is the 3 phosphates. These 3 phosphates. Okay. If you look very carefully at this bond right here is wiggly. Not a straight line, it's a wiggle. Wiggle lines, okay, in chemistry means high energy bond, which means when you break that, you release a little bit of energy. Okay, or more energy than you would breaking other bonds. Okay, so the ATP produced, okay, we use it right away. We don't store ATP at all. ATP is a very big it's too big a molecule for us to use it as storage. In fact, the amount of ATP you use in one day would fill this room. Okay. So you obviously don't do that. Okay. Okay. Oxidation reduction. Okay. So this was actually a really hard concept for me to understand. So we'll put this on a sheet. First of all, the mnemonic oil, right? Oxidation is loss, reduction is gain. Okay. Now here's the thing. Oxidation is a loss of electrons or hydrogens or energy. Reduction is a gain of electrons or hydrogen or energy. This is not really or thing. This is a total thing. So electrons have energy and store energy. So you lose electrons, you lose energy. You gain electrons, you gain energy. Hydrogen just follows electrons around. So anytime you pick up electrons, you pick up hydrogen. Okay. So oxidation is loss of electrons, or H's or energy. Okay. Okay. These reactions are always coupled, so you reduce something by oxidizing something else. Okay. All right. Many molecules can be both oxidizers and reducers in a chain reaction. So I oxidize something else oxidizes me, and I'm reduced in the process. Okay. Oxygen is a great electron receptor, which I'm sure you guys went over in microbiology. Okay. Okay. Usually free electrons not pass along, but hydrogen atoms carrying the electrons are. Okay. So like I said, a molecule that loses hydrogen is oxidized. A molecule that gains hydrogen is reduced. Okay. So some of the stuff we talked about. Okay. NAD and FAD. NAD and FAD is the oxidized form. FADH2 and NADH is the reduced form. So NADH is energetic, NAD is not. Okay? So energetic molecule, no energy molecule. Energetic molecule, no energy molecule. Okay? All right. So you take something will reduce NAD to NADH. NADH will oxidize something else. Okay? Or NADH will oxidize and reduce something else. All right. Hope that makes sense to you guys. And I'll see you guys in class. This is actually recorded before you guys start class. I want to get some stuff done early because I know it's going to be pretty busy once we start. All right. Looking forward to seeing you guys.